My name is Angelina McKenna, and I'm majoring in biology and public health at UMass Amherst, where I perform research in the Cultivated lab. In this lab, we seek interesting information regarding the evolutionary processes of agricultural domestication, and more specifically, the evolution of weedy rice. Through artificial selection, humans have had the distinct ability to mold the characteristics of plants to make them more desirable, creating numerous crop varieties that provide us with the food we eat every day. In inventing agriculture, however, humans also created a new environment of cultivated fields that became available for opportunistic plants, and domestication was thus accompanied by the evolution of unwanted agricultural weeds. Many of these weeds compete with cultivated varieties for resources such as land and water, causing smaller crop yields. In the United States alone, weedy species are responsible for losses of up to $33 million per year. A particularly problematic agricultural weed is weedy rice because it invades cultivated rice fields around the world and competes with cultivated rice, one of the most important crops worldwide. Weedy rice is able to persist and grow abundantly in crop fields because it shatters or drops its seeds to the ground. Seeds lost to shattering remain in the soil and sprout in the next growing season, further infesting crop fields. Because the weedy and crop variety are so similar, it is difficult to manage weedy rice without causing harm to the crop. And currently, very little is known about the genes that regulate shattering. Previously performed genetic mapping in the Cultivated lab has revealed that the gene most likely lies within a region of chromosome 2 that contains 46 potential candidate genes for the shattering trait. Through a multi-step method known as Quantitative Trait Locus mapping, it will be possible to narrow down the number of candidate genes. Mapping begins when one weedy parent and one crop parent are crossbred, and the offspring are then self-fertilized for many generations. The phenotypes, which are the physical characteristics of each individual plant, will be assessed based on the amount of force it takes each seed to be released from the plant. Plants with seeds that require a relatively large amount of force will be classified as having the crop phenotype, whereas those that require only a small amount of force demonstrate shattering and are classified as weedy. Next, we determine genotypes of each offspring. A genotype refers to the genetic information that codes for the observable phenotype, like a set of instructions. By genotyping the offspring, we can see whether they have genetic information from the weedy parent or from the crop parent at various places along the region of chromosome 2 where we believe shattering to be regulated. Places in the DNA where we genotype are referred to as markers. Genotype and phenotype information will then be compared and analyzed. A principle of QTL mapping, due to complex processes involved in self-fertilization, is that the closer to the gene locus a particular marker is, the more similar the phenotype and genotype data of each individual will be. Ideally, using this method, it will be possible to ultimately identify a gene responsible for the shattering characteristic. Not only will this knowledge help to fill in the gaps of how weedy rice evolved, but it could allow weedy rice to be eradicated independently of the crop. This would increase crop yields, save money, and produce more food for the world's growing population. Thank you.